In this activity, I will draw the process of tryptophan attenuation, and you can follow along drawing it as well. In E. coli cells, there is a section of their circular DNA that codes for the production of the amino acid tryptophan. This trip operon can be turned on or off depending on how much tryptophan is available in the environment. First, I'm going to review the simple type of operon regulation we are familiar with. RNA polymerase binds to a promoter on DNA to begin transcription. This operon has a repressor that acts as an on-off switch, followed by genes that code for enzyme subunits that can make tryptophan. Tryptophan acts as a co-repressor, so when it is present and fills the active site of the repressor is bound to the DNA and the RNA polymerase cannot attach. When little to no TRP is present, the repressor is released from the DNA and RNA polymerase is able to bind and begin transcription. Now let's move on to attenuation. After the operator, but before the genes that code for proteins, there is a section of DNA we haven't talked about. The first part of this sequence encodes for the leader peptide, which contains 2 TRP codons, and the next 3 sections make up the attenuator. As you know, bacteria begin translation before transcription is complete. The ribosome begins translating the leader peptide sequence. In low TRP conditions, the ribosome will stall when it reaches the TRP codon because there are not many charged TRP tRNAs available. Because translation slows down, a hairpin forms from the second and third section of attenuator mRNA. This is called the anti-terminator hairpin because it allows RNA polymerase to continue transcription onto the enzyme coding genes. When tryptophan levels are high, translation happens fast, easily translating the TRP codons in the leader peptide. In this case, 2 hairpins are formed. The second hairpin, formed by the third and fourth sections of mRNA, is called the terminator hairpin because it ends transcription before the enzyme subunits are produced. The ribosome disconnects from the mRNA and stops translating, and the RNA polymerase falls off of the DNA and stops transcribing.